Watson and Crick and their DNA discovery. In 1950, Irwin Chargaff discovered the base composition in DNA. Adenine pairs with thymine, and guanine pairs with cytosine. Adenine and guanine are purines. Cytosine and thymine are pyrimidines, as well as uracil. However, uracil is only found in RNA. These bases are most distinguishable because purines have 2 rings, and pyrimidines are composed of 1 ring. In 1953, Watson and Crick proposed a new DNA model. Using Chargaff's findings, they concluded the base pairs between 2 strands of DNA created a double helix structure. Let's take a closer look at the DNA shown on the right. DNA is made up of nucleotides, which are made up of a phosphate group, a 5-carbon sugar, and a nitrogenous base. The phosphate group is demonstrated by the circle attached to the 5' carbon. The 1' carbon attaches to the base, as seen above. In this specific example, the one strand of DNA contains adenine and guanine, and consequently pairs with the other strand of DNA containing thymine and cytosine. The 2' carbon distinguishes RNA and DNA. In this example, the 2' carbon contains a hydrogen, thus it is negatively charged DNA. If this was RNA, the 2' carbon would contain an oxygen and a hydrogen. The 3' carbon binds to the next 5' phosphate. These nucleotide pairings create the DNA helix shape. Base pairs are bonded through hydrogen bonds. A and T require 2 bonds, and G and C require 3 bonds. Base stacking maximizes hydrophobic interactions. These interactions are the main stabilizing force of the double helix. The backbone of DNA is hydrophilic, or water-loving. Additionally, the 2 strands of DNA are antiparallel. Phosphodiester bonds connect the sugar-phosphate backbone. The 5' phosphate group pairs with the 3' OH group of the adjoining nucleotide. One strand of DNA proceeds in the 5' to 3' direction, while the other proceeds 3' to 5'. DNA has major and minor grooves. These are simply where the backbones are farther apart or closer together. It is easier for DNA binding proteins to interact with the bases on major grooves because there is more room. There are various forms of DNA. DNA structures are dynamic, meaning they can become more or less compact. However, they cannot change the direction of their coil. While zDNA is less stable, it is often portrayed in news, advertisement, pop culture, etc. zDNA is distinguishable because the DNA coils up and to the left. Thank you for watching. I hope you learned a lot about DNA in the Watson and Crick model.